Hello, my name is Michael Morikone, and I'm here to talk about my dissertation research, which was on convolutional neural network-based gene prediction using buffalograss as a model system. So buffalograss is a native prairie grass species to the Great Plains region. Swapping buffalograss with traditional turf grasses would help for things such as water conservation, as buffalograss is adapted to the environment better. As a non-model organism, buffalograss lacks genomic resources, including things such as a reference genome. Now, due to the lack of suitable genomic resources, things such as de novo gene prediction can be used to better understand buffalograss. So, de novo gene prediction is traditionally done using hidden Markov model. Hidden Markov models make strict assumptions regarding the independence of states, where any given state is dependent only on its immediately prior state, while independent of all previous to that. Now, there is a concept called synteny, where you have conservation in gene order and gene location across evolutionary lineages. And this concept ensures that this Markovian assumption of independence is not actually met, therefore making hidden Markov model-based methods non-optimal for gene prediction. Gene prediction as a whole is relatively complex in eukaryotes, as we have things such as introns, repetitive elements, and large genome sizes with relatively low genic content. Neural networks have shown in particular to have higher accuracy when leveraging large complex data sets. And in omics work, this has been shown to be upwards of a 40% increase in accuracy in comparison to hidden Markov models. Convolutional neural networks in particular are promising for genomics due to the spatial dependence found in gene location and gene order. And so as such, I developed a gene prediction tool named GeneCNN that uses a convolutional neural network-based approach for making gene predictions. And so in training on the buffalograss genome, training performance reached 97% and validation performance for accuracy was 94.5%. It was stopped at 20 epochs to reduce overfitting. This also led to a F1 score of 0.948. Correctly identifying genic sequences had 97% accuracy, and correctly identifying non-genic sequences had 92% accuracy. To further evaluate the performance of GeneCNN, it was compared against 3 hidden Markov model-based tools using a specified section of the buffalograss genome. And so in this section of buffalograss genome, predictions were made by GeneCNN and compared to the predictions made by the other 3 hidden Markov model-based tools. Predictions made by GeneCNN that were not found in any of the other tools are collected in this column, making them unique predictions by GeneCNN. To further evaluate, the unique predictions by GeneCNN were run against with BLAST to the RefSeq non-redundant protein database to find the level of support for these unique predictions to ensure that related organisms or known protein sequences are actually getting hits from these predicted genes. And as you can see, 80% on average of the predicted genes by GeneCNN that were unique, so not found in the other tools, did actually have hits to the protein database. And so therefore these are likely valid gene predictions that are not being captured by the hidden Markov model-based tools. Next, I looked at the prediction performance in comparison to the other tools to determine the level of support between any given tool combination. As you're going to have algorithmic differences between different gene prediction tools, the vast majority of gene predictions made by these should be shared. And so you can see in single pairwise comparisons between Augustus, Breaker3, and Efganesh, ranged in support of 49 to 84% with GeneCNN. Combinations of 2 hidden Markov model-based tools ranged in support of 41 to 59%, And combining all three hidden Markov model-based tools and looking at the level of support was 40% with gene CNN-based predictions. And so to conclude with, gene CNN performed well as a first-generation algorithm compared to commonly used hidden Markov model-based tools, where it achieved 97% accuracy on test data in correctly identifying genic sequences. 80% of unique predictions made against the hidden Markov model-based tools were supported by the RefSeq non-redundant protein database, making these likely valid predictions that were missed by the other tools. And lastly, when looking at shared support between the different tool sets, we see a range of 49.1 to 84% of all genes predicted by gene CNN that were supported by at least one hidden Markov model-based tool. And so given these results, the field of gene prediction should really reevaluate using hidden Markov models, as methods such as convolutional neural networks are not only more statistically sound, but they also have competitive or improved performance. And so with that, I would like to acknowledge my PhD committee, as well as our collaborators and source of funding. Thank you.